Hey guys, welcome back. This time around we're going to take a look at what myelin sheaths and neurolemas are, and we're also going to take a look at classifications of neurons. Let's start with myelin sheath. Many of our neurons, especially the largest diameter ones, are covered with this whitish protein lipid segmented sheath or wrapping of protein lipid based components called myelin. Since it's lipid based, mostly nonpolar. Now, myelin sheaths are associated with the axons only. They will not be found on dendrites. They act to protect and insulate the axons while increasing the speed of the nervous impulse being transmitted down that axon. Myelinated fibers conduct action potentials rapidly. Unmyelinated fibers are comparably slower, with the difference being upwards of 150 times. In the peripheral nervous system, the myelinated sheath is produced by Schwann cells. Now, these Schwann cells form concentric rings, wrappings, as you can see from this diagram, and they wrap tightly. The more concentric rings there are, the tighter it's going to wrap. This tight wrapping results in lacking any channels or any carrier proteins, making this whole surface nonpolar. That means that electrical vent that's coming down that axon along the axillema, when it hits this region of the Schwann cell and the myelinated sheath, it will have to skip over it because the electrical vent itself is a polar event, and that polar event cannot integrate into that nonpolar myelin sheath. So if you notice from the diagrams, there's going to be spaces in between the myelin sheaths of the peripheral nerves, and these spaces are called nodes of Ravere, and we will see the electrical vent skipping from node to node, meaning that we have to have less depolarization to get from point A to point B. The myelin sheath, again, is nonpolar, a characteristic that makes it an exceptional electrical insulator. Now, before we get into looking at the neurolema and axillema are typically two terms that get confused by students during this part of the discussion. The axillema is always going to be the plasma membrane of the axon. The neurolema is going to be the outer surface of the Schwann cell. Because if you take a look here, you will see that as the Schwann cell makes myelin, the Schwann cell stays on the perimeter and the myelin wraps deep along the surface. It is the myelin sheath that allows the skipping to occur. So the outer lining of the Schwann cell is the neurolema. That would be true also for the outer lining of the oligodendrocyte that would be found in the central nervous system. This neurolema in the peripheral nervous system is created again by the Schwann cells. As the Schwann cells are being pushed to the external, as that myelin sheath is going to be wrapping deep, these Schwann cells will form what is known as this neurolema, or the sheath of Schwann, with the adjacent cell not touching, forming gaps, and these gaps we know as nodes of Ravere. This is an ideal sites for collaterals to emerge, and is the sites where the action potential skips along the axillema. While the neurolema is composed of myelin, the axillema is actually the plasma membrane of the axon. If Schwann cells are not enclosed, recessed neurons are unmyelinated, and the nerve impulse must be propagated the entire length of the axon. Now, this really plays out if you take a look at somebody who has multiple sclerosis. Multiple sclerosis is a disease in which we see a demyelinating of the peripheral nerves. Demyelinating means digesting away of that myelin sheath. And when that digestion away of that myelin sheath occurs, what is happening there is we are now having to conduct that electrical vent, that action potential, the entire length of that axon, instead of having that advantage of skipping from node to node. So that means more of those sodium-potassium channels will have to be depolarized for us to have that electrical vent conducted down the entire surface. So somebody with multiple sclerosis can still have the muscle contractions, the skeletal muscle contractions that they would have had without MS. but they're going to be at a much more gradual, jerky kind of motion instead of the smooth motion that we usually account for muscle contractions. In the central nervous system, the Schwann cells are replaced by oligodendrocytes, and they produce the myelin sheath. Now, if you recall from oligodendrocytes, though, the cell itself is not committed to a single axon. Instead, the cell body stays outside and sends cytoplasmic extensions to a number of neighboring axons. For this reason, there'll be no neurolema associated with the myelinated axons in the central nervous system. The nodes of Revere will be further apart, and the cell nucleus being found not associated with a particular neuron means that if that axon is damaged, that oligodendrocyte will just cut its losses and retract its myelin and redistribute it elsewhere, meaning that that axon doesn't have a chance to grow back through that tube protection. This is an important feat to think about because this myelin is directly related to how effectively can we regenerate a severed nerve. A severed nerve can only be regenerated if it has something there to help guide it to its effector organ again. That something in the peripheral is going to be these Schwann cells. These Schwann cells are not dependent upon the existence of that axon to be there. So if that axon gets severed, it will decay or degrade from the point it's severed to the effector organ. And it'll have to grow back from that severed point to that effector organ. And it will use the Schwann cells that stay there as sort of a tube-like structure to protect it, support it, and guide it through that process. And as long as there's no scar tissue from the point of severing to the point of the effector organ, that neuron given time will grow back. That's not true of the central nervous system because it doesn't have the protective, supportive Schwann cells in the central nervous system. It has the oligodendrocytes that will just cut their losses. Finally, neuron classification. Various ways to classify neurons, but one of the ways is structurally. Structurally, they can be multipolar. In a multipolar neuron, we would find numerous dendrites in one axon. In some instances, though, in the central nervous system, the axon is going to not be present. Then there's bipolar, in which the axon and the dendrite are found in opposite directions. This is frequently found in special senses only, such as the retina and the olfactory. And then there's unipolar, in which we'd see a single process that emerges from a cell body, divides into a T-like projections, some proximal, some distal fibers, with the central process being the axon and the peripheral processes are the dendrites. Now functionally, we've talked about a variety of different types of neurons. So functionally, how do these structural classifications fit in? We've talked about sensory or afferent neurons are typically unipolar, except for the bipolar special senses. Cell bodies are going to be located in ganglia. Remember, that's in the peripheral nervous, with their axons carrying an impulse from the cell body to the central nervous system. Motor neurons are the efferent pathway, are typically made of multipolar neurons. With the cell bodies located in the central nervous system, they carry impulses from the central nervous system to the effector cell via an axon, where they'll form a neuromuscular junction. Now, there's one type of neuron we haven't spoken of that really makes up the vast majority of the neurons. It's called association or interneurons. Now, the association neurons are going to be mostly multipolar. They're going to be confined to the central nervous system. They're going to function as intermediates between the sensory and the motor, especially with where we would find complex integration occurring. And they make up over 99% of all the neurons. But these are also going to be associated with our short-term or long-term memory. If you had psychology and you talked about pruning away neuronal ends, unused neuronal ends, this would be association neurons. If we talk about growing the neuronal net or network as you learn, that would be association neurons. Next time around, we're going to be jumping into neurophysiology. Hey, you guys have a great time. Have a great day. Be safe. See you in neurophysiology.